Hello, my name is Wyvolyn Kirkland, and I will be discussing polymerase chain reaction. PCR stands for polymerase chain reaction. It is a method used to focus in on one segment of DNA and copy it a billion times over. This is a way to zoom in and look at only the thing you need to see in a DNA sample. PCR can be used in numerous ways. It can be used in things like medical diagnostics to diagnose diseases and identify bacteria and viruses. It can also be used in forensics to match criminals to crime scenes. There are 300 billion base pairs that make up the human genome. Using PCR can isolate a specific segment of DNA, but since this is only one piece of the genome, the segment needs to be replicated in order to have enough to work with. In a matter of hours, almost 100 billion copies of a small segment of DNA can be generated. The procedure simply requires some test tubes, reagent, and a source of heat. To begin the procedure, you extract DNA from a small sample. This can be your saliva, skin, blood, or hair follicle. The extracted sample is placed in a PCR test tube, which works by repeatedly heating and cooling the solution, which produces an even heat distribution. The second step is to add 2 primers to the PCR test tubes, which are designed to match the segments you are copying. They attach to either end of the segment of DNA, with one at the top and the other at the bottom. They are very effective because they are unlikely to target the wrong site. Primers can be ordered online, and when doing so, you have to pick a sequence of primer that is specific to the reagent you want to copy. Nucleotides are then added to the PCR test tube. The nucleotides are the building block that the DNA molecules are made of. The nucleotides consist of a mixture of A's, T's, C's, and G's. The DNA polymerase grabs the floating nucleotides and attaches them to the ends of the primer. DNA polymerase is lastly added to the PCR test tube. DNA polymerase copies a cell's DNA before it divides in two. The DNA polymerase attaches to a primer that is base paired with a piece of DNA and starts to add the nucleotides, creating DNA copies. The DNA polymerase most often used in PCR comes from a strain of bacteria called Thermus aquaticus, since DNA polymerase in our bodies can break down at temperatures below 95 degrees Celsius. The PCR test tube is placed in a thermal cycler, which can heat and cool the test tube at certain temperatures at specific times for an hour. The changes allow the reaction to occur. For cycle 1, the thermal cycler heats up to 95 degrees Celsius, where the DNA double helix separates, creating 2 single-stranded DNA molecules. The sample is heated because we don't have any enzymes like we do in the cell to separate the double helix. Then the sample is cooled to 50 degrees Celsius, where the primers lock into their targets before the DNA molecules can try to rejoin. The thermal cycler then goes to 72 degrees Celsius, which activates the DNA polymerase and begins to add complementary nucleotides onto the strands. It falls off when it reaches the end of the strand because it would not have any remaining strand to copy. For cycle 2, the same process is repeated. The desired product is produced in the third cycle. These are DNA copies of the segment of DNA targeted, with 2 strands that begin with primer 1 and end with primer 2. With increase in cycles, you will eventually have over 1 billion fragments that contain only the targeted sequence. Roughly 35 cycles would be a typical length for the amount of cycles to do in your reaction. With each cycle, the strands double, and you can see this with this picture. By the second cycle, you would have at least 4 copies of the fragments, and by the third cycle, you would have roughly 8 copies of the fragments. Thank you.